So I'm here to talk to you about RHD Boost program, which is the program that I've been working on for the last 4.5 years, and it's about improving the biological control of rabbits by looking to introduce another strain of RHD, or Calicivirus as most people know it. Change the slide. So I'm going to start off with a little bit of history about rabbits. Now, the red bit up the top there is where rabbits originally come from, and the purple parts are where they are feral or introduced, and so we've got a really good population. We're the world's resource for rabbits, I suppose. So if they ever go extinct anywhere else, then they'll be safe here. So lucky us. Yeah. Okay, so a brief history of rabbits in Australia. So they introduced in 1859 to Barwon Park in Victoria, just where that arrow is pointing roughly. Thomas Austin introduced 24 rabbits, wild rabbits. There were already small populations of domestic rabbits that were feral, but he successfully introduced wild rabbits. 7 years after the introduction of those 24, they shot 14,000 for sport on his property alone. So, a bit of a problem already. Within 70 years, they'd colonised 2/3 of the country, 5.3 million square kilometres. Fastest rate of spread of any pest animal anywhere in the world. Most definitely facilitated by human beings taking them for food through their travels and just spreading them around. So we helped them out. Significant environmental damage. This is Phillip Island in the middle there before rabbits were eradicated. So there's just that one pine tree just hanging around. Social damage as well. Obviously, a lot of people had to leave the land just because of the impact of rabbits. And so in terms of managing rabbits, we've thrown everything that we possibly can at rabbits, and the only successful way of managing rabbits to date has been through biological control for that broad scale management. Yep. So why are rabbits so successful? Well, females, or both sexes, are sexually mature at 3 to 4 months of age. As soon as that female falls pregnant, she's pregnant for about a month. As soon as she gives birth, she can fall pregnant again. Her first litter will be small, probably 1 to 2 animals. By the time she gets to 10 months of age, she can produce litters of up to 10 animals. So if she's in a place where there's a good season, maybe 4 to 7 litters a year, 10 kittens every litter, doesn't take very long for 2 to become many. So this little diagram up here is based on, they did a control program somewhere in Canberra, I believe, and it was a fenced area, and there were 2 left behind. Just so happened male and female, and it doesn't take very long for 2 to become 184. And then you've got a rabbit problem. You've just wasted all of your time and effort because you didn't remove those last few animals. So the current estimate is that rabbits cost around $200 million in lost agricultural production every year. And then we spend a further $7 million on top of that. And then farmers also spend, just farmers alone, or land managers, another $200 million a year on managing rabbits. So they cost us quite a bit of money. As I said, the most successful control of rabbits has been biological control, so myxomatosis in the 50s and then RHD in the 90s. And there was a recent publication where they looked at the cost benefit of that, and it's estimated about $1.6 billion is what those 2 viruses have saved in lost production over 60 years. So it's pretty significant. We don't know the impact of rabbits in terms of monetary value on the environment because we just don't have a figure for that, but you could expect that it's billions of dollars worth of impact on the environment. Okay, so currently management of rabbits is biological control is the first and foremost, and the 2 viruses still move through the population of their own accord, so they will still turn up on occasion at properties, and then you can also do a release as well. So those arrivals of biological control are a free tool, and then conventional controls are still applied, and the CRC and most people who do research on rabbits and a lot of land managers are aware of this as well, that you have to apply more than 1 tool to rabbits. 1 tool alone will never work. Virus alone will never work. It's just not the way viruses work. Viruses need a host. So a virus is never going to kill that last rabbit. So if you want to get that last rabbit, you have to apply something else. And that may be baiting, or it may be shooting, it may be ripping, whichever tool is most appropriate for where you are. Okay, so this is an example. This is done in Hattah-Kulkyne National Park in Mildura, New Mildura. This just shows you the advantage of applying more than one tool. So the pink line was where they didn't do anything, just so we had a baseline of what was happening with the rabbits. The yellow line is the threshold level. Now for the national park, the threshold level is less than half a rabbit per hectare to allow for regeneration of native vegetation. On production area, that might be different because there was a different cost benefit associated with that. So the blue line is where they just ripped. So they didn't apply any other control method, they just ripped. And ripping is, or harbour destruction, is the best way to control rabbits by far. The green line is where they baited first and then they ripped. So two methods. You can see without baiting, you get down below that threshold level, but within about 2 years you're starting to creep back up again and you're going to have to come back in again. But with the baiting, with the second method applied, you can see you're going to get a much longer term benefit out of that ripping program. Now you could replace baiting with RHD. Or Mixa, turning up in your paddock. It knocks down the numbers and you get in and mop them up with the ripping and destroy the harbour, and you get this long-term benefit from that. There's a study done in Bacchus Marsh, 10 years benefit from one ripping program using the right equipment, heavy equipment, long tines, release a virus, do a ripping program, 10 to 15 years worth of benefit. I have a landholder that I work with. He ripped in 1997 after they did a whole heap of baiting and virus release, and only now is he starting to think about ripping again. So he also, though, is quite good at helping spread virus around, and I will recommend all of you to do this. Whenever he finds a dead rabbit with RHD, no dead rabbit goes untouched by him. He will pick it up and he'll take it into the next valley and shove it down a hole and spread the virus around that way. He even told me the other day if he sees a wedge-tailed eagle on the ground, and I have this vision of this frail old man chasing after a wedge-tailed eagle, because if that wedge-tail got that rabbit, then that rabbit died of something. It probably died of virus and he wants it. So he'll chase down a wedgie to get that rabbit and shove it down a hole in a different valley. And he has very good knockdown with virus on his property just by moving dead carcasses around. So
that's fleas.
A bit of flies. Flies spread RHD, bush flies and blowflies. Yeah, fleas will play a role in that. The way RHD works, and I'll talk about it a little bit more later, but you need that carcass. That carcass is full of virus, and that's how the virus is spread around. So you need a dead rabbit that's died of RHD, full of virus, and flies will land on that. If it's in the burrow, other rabbits, when they use the warren, will step over that and then groom themselves and pick up the virus that way, and that's how an outbreak is created. So you need the vectors and you also need the rabbits. So it's easier to initiate an outbreak in an area where there's warrens than when you've got surface dwelling rabbits, because it's those, we call them virus bombs, those dead rabbits. So next. This is just to show the benefits of the of the biological control programs. So it just shows you also that biological control is never going to be the only tool that you need. So Mixa was released in the 50s. It was estimated about 1.4 billion rabbits at that point, just after Mixa. So in the 1920s, when the major plague of rabbits was on, it was estimated there were 10 billion rabbits in Australia. That's more rabbits in Australia than there are people in the world today. It's a lot of rabbits. So Mixa came in, there was a bit of a crash. Mixa came in and knocked the rabbit numbers down to below that threshold, below that roughly half a rabbit per hectare in some areas, in many areas. Some places there was a 99% reduction in population. But as time goes on, the virus becomes less virulent. There's a bit of a race between the rabbit and the virus out competing each other. Rabbit numbers start to increase again, and then in 1995, RHD escaped, and then in 1996 we let it go. and we saw the rabbits fall down again, and again in some places 99% knockdown of rabbits. So it was around 60 to 90% knockdown of rabbits, but it's on the increase again. Next. The CRC has a number of biological control programs in the works for RHD. So there's the Boost project, which is the one I'm talking about today, the Accelerator Project, and the bioprospecting, which is looking for new diseases of rabbits. But even with those controls alone will never be enough. If we apply conventional control to those, then we can get the numbers down again. So I guess this is just to reiterate that it's a multiple tool approach to rabbits. So the Boost program, the reason that we're here. So in 1995, the Czech strain, which is the only strain that we ever release in Australia, escaped Wardang Island, and then 1996 we started releasing it. Now it's what we call a classical strain. It's one of the first strains that turned up. In fact, the Czech strain is the first strain that turned up. It wiped out the rabbitries in China, 12 million rabbits in 1984. So we've only let the one strain go, but in the rest of the world where rabbits are and these viruses are moving around, there's multiple strains. Now, a strain is just based on where it turns up. So the Czech strain was isolated in Czechoslovakia. You could have a Sydney strain, you could have a French strain, a Paris strain. Sorry?
Like Asian flu.
Yeah. So the rest of the world has these varying strains moving around, and they're competing with rabbits, and there's mixing of genetics and that type of thing. And what we've ended up with is a variant called RHDVA. And RHDVA just means on the outside of the virus, it looks a little bit different to antibodies. So the antibodies don't recognise it, and it can infect the rabbit. So some of the RHDVAs do that and seem to be replacing these classical strains. So they're the ones that we wanted to look at, because we have resistance in Australia to the Czech strain, developing resistance. And I'll talk about each of those types of resistance. So that's what we were interested in, those RHDVAs. Here we go. Okay, so when we talk about resistance to RHD in Australian rabbits, we have 4 different types, and I'm going to talk you through each of these because they're all slightly different. So the first one is genetic. So we know we have a genetic resistant population, and that population is in around the Mildura region. We don't know how far that extends, so I can't tell you if it extends this far, but we certainly know that that hatter population at least, and therefore the surrounding area, is a genetic resistant population. So what happens with the genetic resistant population is that the virus infects the rabbit, passes through the rabbit, doesn't kill the rabbit, but vaccinates the rabbit for life. It has an antibody response and then is therefore vaccinated. So that rabbit can no longer be killed by RHD. And there's a genetic mechanism inside that prevents them from being infected. Next one is an antibody-mediated response, and this usually happens in smaller animals because kittens, particularly under the age of 6 weeks, are less likely to be infected by RHD. The virus doesn't seem to be able to attach. So the virus enters small kittens under 6 weeks of age, and we have the same thing. We have the virus passing through, but essentially vaccinating the rabbit as it goes. So those animals then go on to be immune adults. Maternal antibodies is another way. So the doe who has been vaccinated at some point in her life or survived infection passes those antibodies on to the kitten. Now those antibodies only last for up to 12 weeks, but if that kitten is exposed to the virus in that time, then we have the same thing and they end up with antibodies for life. So those transient maternal antibodies then become permanent antibodies. Now, the last one is RCVA1. Now this is a benign Calicivirus which turned up with the first lot of rabbits, so it's always been in rabbits, and in fact it's now thought that RHD as we know it, that kills rabbits, evolved from this benign virus. So this benign virus has been living in rabbits for a long time, and something happened in it to change it to kill rabbits. So if an animal is infected with the benign virus, while they're infected with the benign virus, if they get exposed to RHDV, there's about a 10-week period while they have these antibodies to the benign virus, they survive infection. That benign virus offers partial protection, so the virus leaves the animal and they're vaccinated for life. So they're the 4 types of antibodies that we're trying to deal with when we're looking for a new strain. So it's no wonder that we're having trouble in some places knocking out rabbits using the current Czech strain. So what we did was we looked at 38 different strains from around the world, and we were trying to overcome all of those antibody types. We couldn't find anything that could kill a rabbit that already had antibodies to RHDV. So all of those antibody types we just looked at, we can't find anything at the moment that can knock those, at least out of the ones that we tested, that can knock them over. So they're still a problem. Thanks. What we could find, however, was a strain that could overcome that protective effects of the benign virus. So that 10-week period. So currently the Czech strain, which I showed you before, if a rabbit's exposed to that while it has this benign virus, it gets vaccinated for life. With the new strain, the K5 strain, which is what we're calling it, we exposed rabbits to the benign virus and then exposed them to K5 and killed them. So that means that wherever the benign virus is, which is generally in the cool, wet regions of the country, all down the east coast, into Victoria, southwest WA, wherever that benign virus is, if an animal is only infected with the benign virus, and there's about up to 25%, could be 0 to 50, but let's just use 25 as a middle ground, you're talking about 25% now extra animals that are susceptible, potentially susceptible to this new strain. It also, the new strain required less virus to kill a rabbit. So, which is a good thing because there's less chance of getting a suboptimal dose. Less amount of virus to kill a rabbit means more virus, could potentially mean more virus available to kill other rabbits. So less virus and killing them and replicating faster is a good thing as far as we're concerned for this. So killed them faster, which is good. So rapid infection and then killed them faster. We don't want them to die too fast because we need the virus to build up. So we create this bomb, but we don't want them to drag out. We don't want that death to drag out. Quite obviously, you know, we're trying to be humane in the way that we manage these animals. So let's get them infected and die, but infected enough so that that virus can then spread through the population and cause an outbreak. And the other good thing about K5, as I mentioned, kittens aren't susceptible at a young age or seem to be less susceptible. And what we didn't want was a strain that was going to infect kittens better. And this strain doesn't. So, and that's a good thing because we don't want our kittens to get infected with RHD. So this was no better than what we already have. So we're not going to make the situation worse by vaccinating lots of young kittens. So can we let this go and can we get it to establish? The short answer is I don't know. We don't know. If we have a look at Mixo, we could never establish a second strain of Mixo in the field. So you could use Mixo, other strains, we've used other strains, and you could knock out a population. But that additional strain would never establish itself and move through the population. It would just wipe out that patch of rabbits and then you would have to reapply it. So the strain that goes through the population now is what was first let go. There are way more virulent strains of Mixo around. We could just never get them into the population, to persist in the population. So if we look at that, then we might say, well, we're never going to establish a second strain of RHD. But the two viruses are very different beasts. So as I said to you, the RHD creates this virus bomb and this dead rabbit that is full of virus. So RHD is geared to building up quickly and killing the rabbit. It needs a dead rabbit to effectively move itself through the population. Mixo works in the other way. So Mixo, if anyone's ever seen a Mixo, never seen a Mixo rabbit, they're quite unattractive. Full of pustules and conjunctivitis, and it's a horrible disease. And the way that it's spread is those pustules. So mosquitoes will come and suck the blood out of the animal and out of those pustules and spread it around that way. So the longer the animal is alive, the more chance it has, the virus has, of being spread. So the virus has changed over time to keep the animal alive for as long as possible. So we're now talking about 4 to 6 weeks before an animal will die from Mixo, and generally they tend to die of something else. So they'll die of pneumonia or something like that. If you've never picked up a Mixo rabbit, there is nothing to that rabbit. So there was one Mixo rabbit that I was trapping all the time, and at the end of the trapping session we were killing our rabbits and taking blood from them, and was dry. I couldn't get anything. There was no meat, no nothing. It was just a shadow of a rabbit. So it's yeah. And we would never let something like that go now. But yeah, so we think based on those differences in the way the virus behaves, we have a pretty good chance of establishing a second strain. And if we look at what's happening overseas, that's going on. So should be good. So where are we going to see the impact of boost? The areas highlighted surrounded in green are the cool, wet regions, high production areas of the country. The red dots are where we've found the benign virus, RCVA1. That's where we expect to see the greatest knockdown of with K5. So the research that I've been doing for the last 4 years, sampling rabbit populations across the country, on average, about half of the rabbits already have antibodies to RHD. So about half of the rabbits are not susceptible to RHD. If we go to these cool, wet regions of the country, it goes up. And you're talking about 60 to 70% of your animals already have antibodies to RHD, of your older animals. So, as I said before, it leaves us with about 25% animals that only have the benign virus, so they're our target animals for the K5. So that's the opportunity, is to increase the effectiveness of RHD in those cool, wet regions. Now, it's still going to have an impact in the drier areas. It may well have an improved impact. We don't know that, but the focus of this is because it overcomes the benign virus. We're really interested in where the benign virus is. So now we're hoping that we can release this soon. We've submitted a registration package to the APVMA, who are in charge of deciding whether you can let this sort of stuff go. We've been advised that we should hear something by July of this year, but I was also supposed to hear something from the APVMA in November, and I still haven't heard anything. So maybe the second half of this year we might hear something back from them. If that's the case and we get the go-ahead, then we're expecting to release in autumn the following year that we get approval. So all things going to plan, autumn 2016, but don't hold me to that. Now, the Boost project isn't as funded as, say, the RCD project was in 1995-96. So there just aren't the people on the ground anymore to do all the work that was done in 1995, or the money. So we're asking for everybody's help. Now, you can be involved in this as little or as much as you like. There's a range of ways that we can get landholders involved. So one way is to just simply download an app, which hasn't been developed yet, but will hopefully be done by the time this. It all hinges on funding. So there'll be an app where you can just basically say, oh, well, I've got RHD dead rabbits on my property. Click on that. It'll use your phone's GPS to map where you are. You can take a photo if you like. We recommend that, because then I can have a look at the picture and go, looks like roadkill, or maybe it's not RHD. And then you can also opt, so you can just do that, as simple as that, just record that you have a dead rabbit. That will link into the rabbit scan, which will have a separate layer on it which will show disease. So you can then have a look at the map of Australia on rabbit scan and see where outbreaks are happening. And then you can use that information. You go, okay, well, outbreaks are starting to occur around my area. I'm going to plan my follow-up control for when that virus arrives. So you can take advantage, rather than just sitting there and then all of a sudden finding dead rabbits and you haven't had time to plan your follow-up. You can use this for that. So the simplest way is just to map dead rabbits. You can then submit samples of dead rabbits so that we can confirm what it was and whether it's the new strain or the old strain. The other thing that you can do, and we're going to be coordinating through CMAs, NRMs, LLS, depending on which state you're in, which body we're talking about, to set up some sites in people's properties. And what we'll be asking for will be do a count of your rabbits, so maybe drive through a paddock for 2Ks or something and do a count, let us know how many you've got. So you do that for 3 nights so we get an average. Then you do a free feed, release the virus, and then 2 weeks later go out and do another count for 3 nights and let us know the difference. If you've got your firearms license and you want to take some samples, then you can shoot rabbits before and after, or at least before, so we know what the profile of your rabbit population looks like. And we can provide you with kits for that, coordinated through the CMA. And if you can pick up any dead rabbits that you find as well, then we'd appreciate that as well. We want your dead rabbits. So, so there's many ways to get involved. And if you're super keen, then we can set up a more long-term site where we have sites already established where we monitor every 3 months. So we do a 3-night count and we shoot 20 rabbits every 3 months. We're doing that now before, so we know what the population looks like beforehand, and then we'll do it afterwards. And if you want to get involved in that, then by all means come and see me and we can help set that up. So there are a few rules associated with those, so we'll have to, depends whether your property's big enough and that type of thing. But there are plenty of ways to get involved. Now it's kind of like a giant nationwide experiment. And it's not often that you get the chance to be involved in a giant nationwide experiment. So if you want to get involved, then I would be more than happy to have you